Hello, everybody. We're going to be talking today about cell cycle checkpoints and regulation. We're going to be looking into how the cell cycle control system triggers and coordinates key events in the cell cycle and how it's regulated by both internal and external signals. The checkpoints, based on these signals, will tell the cell either to move forward with the cell cycle or stop until the damage can be repaired. There's going to be three major checkpoints for us. Which will be our G2 checkpoint. Our M checkpoint. Most specifically, we're looking at metaphase within mitosis. And then the third one would be our G1 checkpoint, sometimes called the G1S transition. Okay. Now, we'll be looking at G1 checkpoint first. Okay. And this is really the primary point when the cell must choose to divide. Okay. So there's going to be a few things it's going to check for. Right. The first of which is going to be size. Is the cell actually large enough for division? Right? If we're going to take one cell and divide it into two cells, there's got to be enough stuff for both cells. Number two here, nutrients. Energy is important. If there's not enough energy to do this process, we're not going to go through with it. Three would be molecular signals. Molecular signals. Or that can cells communicate with each other. Okay, are the growth factors present in order for this process to proceed? Next one, we want to look for DNA damage. Is the DNA actually okay? We don't want to start cell division and give one of the cells bad information, bad genetic information. All right. Now, at this point in our cell cycle, okay, by the end of G1, cells that don't pass this, cells that do not pass this checkpoint, often end up in this side phase called G0. And you can kind of notice up above, and I'll just highlight it so you guys can see it a little bit better. Okay, that says G0, and it kind of looks like go, but it's actually the opposite. It's like a pause. Okay, so it's really saying, hey, the cell's not ready yet. We're going to go into this pause phase before we actually proceed. All right, G2 checkpoint is going to be similar. Okay, so we go through this G2 checkpoint after S phase is really going to check again the DNA. Is the DNA damaged? That's not good. If it is, we're going to stop our cell cycle at that point. What's important about this checkpoint is this checkpoint is after synthesis. Okay, so synthesis G2, and then we get to the end of G2 and we have this checkpoint here. So it's important for us to look at DNA replication. Was the DNA actually copied correctly in that S phase? Again, we don't want to create a new cell if it doesn't have all the information, all the correct information that we need for it. The third checkpoint, our M checkpoint, is also called that spindle checkpoint. In metaphase, it's going to check if those spindles are attached. So we're looking for spindle attachment. Spindle attachment. These spindle fibers are going to end up pulling apart our sister chromatids. If that is not attached correctly, the end result is not going to be what we want. So we want to make sure that that's okay there. All right. So our cell cycle is regulated by proteins that control the sequence of the events in the cell cycle. Okay, so it's going to be controlled by a few proteins here. The two primary types are going to be called cyclins and protein kinases. Protein kinases, there we go. All right, we're going to skip down and we're going to talk a little bit about cyclins and protein kinases, and then we'll jump back to our image above. Cyclins are a group of proteins that control the cell cycle, by activating protein kinases. Cyclins are a group of protein that control the cell cycle by activating protein kinases. Each stage of the cell cycle is associated with a specific cyclin. Remember, this is very tightly regulated, so we want specificity within this process. Each one of those cyclins is going to activate, each cyclin activates a specific protein kinase. Okay, there's a specific cyclin for each stage, and that specific cyclin is going to activate a specific protein kinase. Okay, so let's jump up really quickly towards our diagram above. On our x-axis, basically we have time, different phases of the cell cycle. Our y-axis is the concentration of different cyclins. Okay, we should see that the rise and fall with all these different cyclins are going to lead to a transition from one cycle to the next. Okay, so what do we need to memorize about this? Not necessarily where each cyclin is present most of, but knowing that when the cyclin is present, it's going to force it to move from one phase to the next. All right. Let's take a look at our diagram. When cyclin is not present, okay, so our left side here, okay, this protein kinase, the CDK here, is going to be inactive. Okay? These cyclins' purpose is to activate these kinases. Okay, so let's go ahead and jump down. Protein kinases are enzymes that activate inactivate other proteins through phosphorylation. They're going to be often present in constant levels. They are often present in constant levels and it's an active form. Okay, so they're there. They're just not doing their job because they haven't been told to by these cyclins. Okay, So you may have heard me mention before that the attachment from the cyclin really is going to activate this protein kinase, and this is specifically called a cyclin dependent kinase, which is abbreviated as CDK. Okay, so it's just a specific type of protein kinase that, again, is dependent on activation through cyclin. Okay, so the activation of a CDK rises and falls. Rises and falls with its specific cyclin's concentration. So the activation of CDKs will rise and fall with its specific cyclin's concentration. So let's jump back above here. Okay. We can tie together when there is higher concentrations of a specific cyclin, that CDK is more likely to be active. Okay. That's all that last one's saying. And then the activation of a CDK directs the phosphorylation of a specific target protein. Remember that kinases are enzymes that are going to phosphorylate other proteins. Okay, so what this is doing is when the CDK is activated, it's going to end up phosphorylating a different protein to activate or inactivate that one. All right, so let's talk a little about a specific example of this, and probably the most common example when we look at cell cycle and regulation with it. Maturation promoting factor, MPF, triggers the cell cycle to move past the G2 checkpoint into the M phase. Okay, so cyclin B, often called M cyclin because of the phase it's going to transition to, is present throughout the cell cycle, but begins to accumulate in the cell near that G2 M transition. From G2 entering that M phase. Okay, this MPF is really the cyclin CDK complex. Okay, so when enough of this MPF is accumulating, so if enough of it accumulates, it acts as a go symbol. Okay. A go signal at the G2 checkpoint. Okay, so if there's enough MPF there, it's going to say, hey, you can proceed. You can keep going through our cell cycle and enter that phase. Okay, so MPF causes a phosphorylation. MPF causes a phosphorylation cascade that triggers the events of the M phase, or the events of mitosis. Defense of mitosis. Remember that phosphorylation cascade is this very common thread throughout cell communication. So during anaphase, MPF activates a protein that breaks the cyclin down. Allowing the cell to exit M phase or mitosis and enter G1. All right, so let's scroll up back to our image here and kind of take a look at what we're seeing. Okay, so remember that cyclin B is this M cyclin we've been talking about. Okay, we can see here low levels kind of early on, but as the cell cycle proceeds, we're going to see more and more cyclin B here accumulate, this MPF. When we get this cyclin B to accumulate and therefore the MPF to accumulate, it's going to basically tell the cell that it can transition from G2 here to mitosis. And the signal to come out of that is the breakdown of this molecule. So once we decrease that concentration, it's telling the cell it's okay to go from mitosis back into G1. All right, so let's take another look here. Okay, we have the M cyclin. Remember, cyclin B is that same thing. When it binds to our CDK, that CDK is activated, and it's going to phosphorylate a specific target. Okay, so M phase targets for the proteins important for M phase are going to be phosphorylated, and this M phase is going to begin. Okay, it's going to do those things that need to be done. Spindle fibers form, chromosomes are condensing, nuclear membrane breaks down. Okay, so a little bit more of a specific example with that when the nuclear membrane is being broken down. Here's our protein who's going to add that phosphate group to a certain target, and that certain target's going to go ahead and kind of break down this membrane in order to get a little bit more access to that DNA. Okay, I hope that helped. Please write down any questions that you do have at the end of the video, and we can start class by talking about it.